A big hello to all my cute little champions. I hope you're doing great, my dear children. I welcome you all to Masterstroke series, where today we are going to do the next chapter of PU1, that is cell cycle and cell division. Now this chapter also gives you almost 1 or 2 questions each and every year, so this should not be missed. As I told you, so the chapters that skip certain years, I'm going to club them up and I'm going to provide you a video that could be useful for you. I would be mainly solving questions there and explaining the concepts together. Okay, complementary to that. Now, one very important thing is that we are going to bring you something really nice for more question practice. I often see certain comments that, ma'am, are these questions enough? Being a very honest teacher or a human being, I can tell you one thing, that is, KCET is a competitive examination meant for increasing the competition. You get it? So obviously, the questions that I give, the questions that Aishwarya ma'am gives, or Swami sir, or Ankit sir, or any institution out there, my dear children, it would not be repeated as such. There will be so many questions that would be coming your way. What are we doing over here? That might be the next question that you are having. We would be helping you with the concepts. So there are certain concepts which are repeated. While we solve the question, we explain you the concepts, and the same concept can be applied for another question out there. So that is your job, to practice more and more questions. Please do not wait for just the videos and say, if the video is not coming, I'm not going to practice. Please don't do that. I want you to score really good, which is why I'm honestly telling you, keep practicing more questions. We are also going to bring so many questions out there really soon for you guys, so that you have basically our dream is to make you score really good and make you land up in your dream college. So your dream is also our dream over here. So we have a small common factor over here. So my dear children, keep working hard, and all your comments, all your things that you share with us, it means a lot. Whatever doubt you have, okay, suppose you studied elsewhere, okay, you had a doubt in that particular question, please put that doubt over here in the comment section. Okay, at least if I'm talking about myself, I try to reply to all the comments, so I would be replying to your comment as well if you ask me a doubt. And all the good and bad things, whatever you want to say, you can tell in the comment section, my dear children. It's open for you guys. And yes, definitely do not forget to basically share it with your friends as well if you find that what we are doing is useful for you and your friends. Yeah, now let's get ahead over to what is cell cycle. So my dear children, just imagine there's a building. Okay, there's an old building, old worn out building. There are so many people out there. It's very dangerous to stay in there. This building would collapse anytime. At that point of time, what do you need? You need to start basically building what? A new building. In our body also same thing is happening. So many cells are dying each and every day. Okay, they are not good enough now. They worked really hard the whole life. Now it's not fine. Okay, they are tired. They cannot further divide or things like that. So what they are going to do? Basically, they should be replaced by the new cells. And this process of basically making up a new cell, okay, the events which are there in making up a new cell, it is known as cell cycle. The sequence of events by which a cell duplicates its genome, synthesizes other cell components, and divides into 2 daughter cells. So basically a cell is there. Okay, when you talk about a cell in general, it will be having the genetic material, and it will be having what? The organelles. It will be having the organelles, and it will be also having the genetic material. So basically, when it has to make a copy of itself, obviously it has to include this thing and also the cellular components. Over here also it will be doing that. So for doing this, for basically giving their things to 2 other cells, 2 other daughter cells, my dear children, obviously it has to undergo what? Duplication. And after dividing, will this cell be there? No, my dear children. This cell, when it divides, it will be divided into 2 cells. So you would not have this mother cell that you see over here. So one copy is made, and then that's all. So this event that happens, my dear children, is known as cell cycle. What are the processes that is involved over here? So the processes that is involved is cell growth. So there is a cell. Okay, there is a cell. It has to grow in size such that it can divide into what? 2 cells. So cytoplasmic increase happens over here. The DNA replication, I told you, apart from DNA replication, all the organelles which are there, they also have to duplicate. And finally, after duplication, after increase in increase in the content, finally when they think that it's perfect, now they divide, and that is known as cell division. And all of it obviously need what? Coordination. So without coordination, do you think anything can work? No. For example, if my what I am seeing and what I am perceiving does not go together, my dear children, it won't be of any use. You get it? So similarly in our body also, there is very much importance of what? Coordination as well. Now DNA synthesis happens only during a specific stage of cell cycle. I am going to discuss about that and all the other things which is there. So over here, phases of cell cycle. Now the events which are there in the cell cycle, we are going to talk about that, and it's really interesting. You know what? In your NCERT, there are 2 eukaryotic cells example. 2 cells which are eukaryotic. What do you mean by eukaryotic? The cells which have membrane bound cell organelles, and also where the genetic material is inside the nucleus and the nucleus is guided by a nuclear membrane, my dear children. This cell will be known as a eukaryotic cell. For example, me, you, all are made up of what? Eukaryotic cells, which is why we are known as eukaryotes. Apart from us, there is one more chutu guy that is known as fungi, if you know fungi, right? And in fungi, there is an example given in an NCERT, that is what? Yeast. That is yeast. Both of us are going to undergo division now. Okay. So, how much time do you think that our cells take to divide? Our cell basically takes 24 hours to divide. Can you believe it? 24 hours is a big deal, right? And how much time does an yeast cell take to divide? Just 90 minutes. Just what? 90 minutes. 24 hours and 90 minutes. So, 3 hours, sorry. Almost 1.5 hours I can say. Yes. So, 24 hours and 90 minutes, my dear children. Please remember that. That's the whole duration of the cell cycle. Now, in this 24 hours, if they are dividing just into 2 parts, what they are doing actually just to divide, it needs 24 hours? No, my dear children. So again, your 24 hours are divided into 2 phases. One is interphase and the next one is M phase. What do you mean by interphase? Interphase is a phase where the cell is actually getting ready for the division. Okay, and M phase is the one where the cell actually divides. So I am going to tell you one thing. In the interphase, it requires 23 hours, whereas for the M phase, it just requires 1 hour. Total becomes what? 24 hours. So ma'am, 23 hours they are taking for preparation. What is this? What kind of party is this happening? So basically, just like your examination, you guys might have started preparing from 1 year, or 6 months, or 3 months, or probably yesterday. Anyway, at least 10 days you will prepare. At least for 10 days, probably someone would be preparing out there, right? The preparation time and the date of examination, you just have 1 hour for 1 paper, my dear children. That is very less. The actual date of examination, the preparation time, if you see, it's very much. Similarly over here also, in the cell division, my dear children, there is too much time taken for preparation. Why? Because the DNA has to get replicated, the organelles have to get duplicated, the proteins have to be made. There are lots of things in there which is happening in the back. You know, preparation is the most important thing that is happening over there. Okay, which is why interphase is actually known as growth phase as well. Okay, interphase is also known as growth phase, but sometimes it is also known as what? Resting phase. So someone who is there out, okay, who see you all for 1 year, probably there might be repeaters who might be staying at their home studying, but for few they might feel that they are resting. These children, they are not studying anything, they are just resting. But are you actually resting, my dear children? No, you are working really hard. So basically you are in the growth phase or interphase. So interphase is known as resting phase also, okay, and also growth phase. Fine. Now, because resting phase, because cell division is not happening, that is why it is known as resting phase, not because it is actually resting over there. Okay, so it takes up more than 95% of the cell cycle duration. Yeah, you saw 23 hours means 95%, right? Prepares the cell for division through growth and DNA replication. So it is going to grow and also replicates the DNA content. Now, what happens in M phase? M phase is just 1 hour in the 24-hour human cycle, begins with nuclear division, that is karyokinesis. That means, if suppose the cell is ready with the DNA, the cell organelles and everything. Now, what will happen? The nucleus which is there, it is the one that will first start to divide. Okay, it was the first which will be starting to divide, my dear children. The nucleus has to divide into 2, and then later on, the cell which is there, it will also divide into 2. So this is a cell, the nucleus divides into 2, and then later the cell divides into 2. So what do you call the nuclear division as? So I said M phase starts with nuclear division. What do you call this as? Do you have specific term for it? Yes, it is known as karyokinesis. Karyo means it came from carrion and it means nucleus, okay? And kinesis means division. Now ends with cytoplasmic division. What do you call this? Cytokinesis. Cytokinesis. So my dear children, here you have the cell cycle. So from this point to this point, it is the interphase, 23 hours, yes? And this chutko phase that you see is what? Basically M phase. So M phase will be starting with what? Karyokinesis, where your nucleus is dividing, and then it will end with what? Cytokinesis over here. Karyokinesis, again, it will be divided into multiple phases. I'm going to talk about that. Please don't think, oh my god, how many phases are there? Hurry, chill, it's easy, I am here. No, trust me on me. Chalo, let's get ahead. Now we are going to talk about interphase. So interphase is divided into 3 phases. Phases, yes, phases. And it's going to be nice, okay? So first phase is G1 phase. What do you call it? G1. Okay, G1. So what is happening over here? Interval between mitosis and DNA replication. Didn't get it? I'll explain to you later. Cell is metabolically active and grows but does not replicate DNA. So in G1 phase, what is happening? It has started off. So when you start off your preparation, first you open your NCERT, then you make the strategy and things like that. That's how it goes, right? Similarly over here, it is not metabolic. It is what? Metabolically active, as in things are going on in there, my dear children. And the cell is growing in size also. The cell is growing in size also, but it does not replicate the DNA. It does not replicate the DNA. Okay? Cell grows in size and is metabolically very much active, my dear children, but does not replicate the DNA. Now, I want to tell you one thing. In this phase itself, when I say it's growing and it's metabolically active, certain organelles, okay, certain organelles are going to duplicate. For example, mitochondria, for example, endoplasmic reticulum, and if you have chloroplast, in the case of plants, chloroplast also. So all of these things are going to duplicate. Few organelles, okay, few organelles duplicate over here, very few. Okay, so organelles have duplicated, little few of them, and they have grown in size, they are metabolically active, but DNA replication is not happening over here. Then DNA replication happens where, ma'am dear? So ma'am dear, oh my god. So where is it happening, dear? It is happening in the S phase. It is going to happen in the next phase, that is S phase. S phase is also known as synthesis phase. What is getting synthesized? DNA is going to get synthesized. So DNA replication occurs over here, my dear children. Please remember, DNA replication occurs what? Over here. Okay, which is why G1 phase is known as interval between mitosis and DNA replication. So this here DNA replication is happening, and here what was happening? Here mitosis was happening. G1 phase is basically coming between S and the M phase, which is why it is known as, it is coming between mitosis phase and D. So it is known as pre-synthesis phase. It is known as pre-synthesis phase, yeah, and it is also known as post-mitotic phase, because it is happening after mitosis as well. After one cycle of mitosis, the next begins from here, okay, from the G1 phase. I hope you got that. Cool. Now, one more very important thing. So when I say that DNA replication occurs, that means the DNA content is going to what? Increase. It is going to get increased. So my dear children, I have a question for you guys. So the DNA, it was 2C first. After the synthesis phase, obviously it gets replicated. So it will be what? 4C, double the content of DNA is there. What about the number of chromosome? What about the number of chromosome? The chromosome is the one that carries it. Okay, that carries the DNA. If you talk about the number of chromosome, what will be the number of chromosome? If you say that initially you started with 2n number of chromosome, what will be the number of chromosome after the S phase? Ma'am, here the DNA has replicated, so there is one more strand that you see, so which is why this is one chromosome and this is the next chromosome. So it will be, if it was 2n, double it will become 4n. There you get things wrong. My dear children, if you see, it is attached over here with the help of a centromere. What do you see? This is a centromere. And when you basically, for now counting the number of chromosome, you count the number of centromere. Here also it was 1, here also it is 1. Even though the DNA which was there, it got duplicated and you have a new DNA strand over here, it is still attached. Okay, it is still attached with one centromere, which is why you will say if it was 2n over here, it is still 2n here also. It will be still 2n here also. What do you call this as? These together they are known as sister chromatids. They are what? Sister chromatids, my dear children. So the DNA, always remember, the amount of DNA, if you ask, it is increased or not? Yes, it is increased. If it was 2C, it becomes what? 4C. But what about the number of chromosome? Baba, it remains the same. It was 2n, it will be remaining 2n itself. Will you be able to remember it? You have to remember it, okay? So, if DNA was 2C, it will increase to 4C. Chromosome number remains the same, that is 2n, remains 2n. Very important. Now, in animal cell, DNA replicates in the nucleus. Agreed? Okay, DNA will be replicating in the nucleus in the case of animal cells because we are eukaryotes, right? Centrioles duplicate in the cytoplasm. That means what, my dear children, along with DNA replication, one more thing is happening, that is centriole is also getting duplicated over here in S phase. So two major things happens here. One is DNA replication, other one is what, centrioles also get duplicated. Centrioles are the one that will be giving rise to what, spindle fibers. That will be giving rise to spindle fibers, and that will be taking away. So for example, you have the chromosomes lined up somewhere, and you want to separate the sister chromatids. You need to attach spindle fibers which will be separating it. So who is the one that will be producing this spindle fibers? It will be centrioles. So centriole duplication as well as DNA replication occurs in the S phase. Please remember it. So in the G1 phase, they are metabolically active. Growth is very much happening over here, and also few organelles are getting duplicated. Then DNA replication happens in the S phase, and also the centrioles also are getting duplicated in the cytoplasm. So S phase, two things are happening. One is happening inside the nucleus, that is DNA replication. One is happening outside the nucleus, that is in the cytoplasm, that is centriole duplication. Please remember that. Okay. Now, in G2 phase, some children know, right, centrioles are also getting duplicated in the nucleus. No, centrioles are getting duplicated in the cytoplasm. Please remember that. Okay. Now, G2 phase, the cell prepares for mitosis by synthesizing proteins. What? It will be synthesizing proteins, my dear children, and the growth continues. Over here again, there we duplicated few organelles. The other organelles, my dear children, whatever leftover organelles were there, they are going to duplicate over here. The organelles duplicate again over here. Whatever leftover organelles were there, they will be duplicating over here, and one very important protein duplicates over here, that is known as, synthesized over here, that is known as histone proteins. Histones are the one that basically make the DNA compact, the chromatin compact. Okay, so histone proteins are also duplicated over here. So I hope you got this. Okay, in interphase, the most amount of time is taken by G1. Okay, G1 takes the most amount of time. You might think S phase takes more amount of time. No, it is G1 phase. Now, there is another phase apart from G1, S and G2. That is this phase. You call it as quiescent stage or G0 phase. Okay, so some adult cells, example heart cells, do not divide. So what happens? They are like, I cannot divide, I cannot undergo this much thing, I am already helping in the pumping of heart, I cannot undergo cell division, let me just rest over here. I will be metabolically active, I will be doing my own work over here, I am not dead, okay, please do not consider me as dead, I am very much metabolically active, but I am not going to divide. I am not going to divide. Please make it very clear, okay. So some adult cells such as heart cells do not divide. Some cells divide only when needed, that is to replace the lost cells. So some cells are there which will be only dividing when some other cells are not there. So all of them, where are they basically going to stay, my dear children? They are going to stay in the resting stage or G0 stage or quiescent stage. So these cells exit the G1, okay, they would be exiting the G1, they do not want to undergo all the division, and they will be in the G0 phase. They are metabolically active, please remember that, but they stay metabolically active, they are not dead, okay, just that they do not divide. Fine, cool, works. So interphase, G1, S and G2. Apart from that, you have G0, this is known as quiescent phase or resting phase. Do not think that interphase is known as resting phase and this is also known as resting phase. No, basically resting stage. Okay, G0 is known as resting stage or quiescent stage. I hope that is clear. Cool. Now, we are going to talk about this part, that is the M phase. So M phase, my dear children, it is the most dramatic phase of the cell cycle because here things are happening. Okay, so cell is finally going to divide. The ultimate exam battle is going to come over here in this particular thing. Okay, so it involves major reorganization of all the cellular components. Since chromosome number remains the same in the parent and daughter cells, it is also known as equational division. Okay, in the case of M phase or mitosis, you are talking about mitosis over here. Okay, so mitosis is known as equational division. Why? Because as I said, we start off with a cell that has 46 number of chromosome, we end up with a cell that will be having 46 number of chromosome itself. Okay, which is why we call this as an equational division. The number of chromosomes remain the same. Now, karyokinesis or nuclear division. I told you in M phase, it starts with what? Karyokinesis. So mitosis is divided into 4 stages, but it's continuous process. Okay, so please remember, all the stages that I'm going to talk now, it is going to be continuous. It's not like, okay, this step is done, that means this stage is over. It's not like that. There is no clear distinction between the stages. The clear distinction between the stages are not always visible. That is what it meant. Okay, so we are going to start with M phase. where we have karyokinesis. The karyokinesis is actually divided into 4 phases. One is prophase, then we have metaphase, anaphase, and telophase. There is no clear distinction between these phases, my dear children. Cool? Now, prophase. What happens in prophase? So, cell has everything, duplicated organelles, the DNA, everything. So, new DNA, it follows S and G2 phase, I hope you know that. So, new DNA formed from S and G2 phases are intertwined and not distinct. Okay, they are intertwined, they're coiled, and it is not distinct. Okay, so chromatin condensation begins over here, leading to formation of compact mitotic chromosome. So chromatin fibers are like this bead on the string-like structure that you have, my dear children. We have histone proteins onto which basically the DNA is coiled around like this. But we have to make it still compact. Okay, we have to make it more compact. So chromatin which is there, it undergoes more condensation. Okay, it begins more condensation, leads to the formation of what? Compact mitotic chromosomes at the end. Each chromosome consists of 2 chromatids joined at the centromere. So as I told you, it made a copy. Now you have a centromere. This is known as centromere, dear children. Okay, the centrosome duplication which occurred in the interphase. Centrosome duplication means centriole. Okay, centrosome has centrioles, my dear children. So basically centrosome duplication happened in what? The S phase itself is now visible. The centrosome is now visible. Okay, and centrosome begins moving towards opposite poles. So if this is a cell, oh my god, a cell with this big chromosome. Just a second, okay. Suppose this is a cell. What happens? The centrosome which is there, probably somewhere over here, they would be starting to move towards the opposite poles. They would be starting to move towards the opposite pole, one over here and the next one over here. So that happens over here. Each centrosome radiates microtubules called asters. What are they going to radiate? They are going to produce some microtubules called what? Asters. The mitotic apparatus is formed consisting of 2 asters and spindle fibers at the end. Okay, so they moved there. Okay, now what is happening? Basically, I told you that centrosome are going to produce what? Microtubules, that is known as asters. So they are over there going to produce 2 asters and also spindle fibers. They are going to produce 2 asters and also spindle fibers. Okay, so I'm going to sum up and tell you what are going to be formed over here. So first thing, chromatin which was there, okay, it has to condense. So it started condensing, it begins. Okay, chromatin condensation begins. It's not like it ended over there. Okay, so chromatin condensation begins, leading to formation of what? Compact mitotic chromosomes that you see. Now after that, centrosome that is duplicated, my dear children, it has started to get visible, and now it has started to move towards opposite pole, and they made something. What did they make? They basically radiated asters. Okay, they radiated microtubules, what? Known as asters, forming 2 asters. They have 2 asters over here and also spindle fibers, and also what? Spindle fibers. Now cellular changes at the end of the prophase. What all cellular changes are there? The Golgi complex, endoplasmic reticulum, nucleolus, and the nuclear envelope, it will disappear. So this was all happening inside the nucleus, my dear children, but now the nuclear envelope and everything has disappeared. There is no nucleolus also. There is no nuclear envelope. There is Golgi apparatus, endoplasmic reticulum, all have started what? Disappearing. All have started disappearing over here at the end of prophase. Okay, now something big is gonna come. Yeah, something big is gonna come. That is why. So I hope you remember all these key terms that are being put. Okay, chromatin condensation, centrosome moving to the opposite poles, formation of aster, and the spindle fibers, that is the mitotic apparatus. It is known as mitotic apparatus, my dear children, and the Golgi apparatus, endoplasmic reticulum, and nucleus and nucleolus going away. So it is, you can remember it as Gen. Okay, you guys are Gen Z, so over here it's just Gen. Okay, so Golgi complex, endoplasmic reticulum, nucleolus, and nuclear envelope, they started disappearing. Now we have also, so this is the thing that you have. So initially it was like this, my dear children. Slowly you see the chromatin fibers have become the chromosomes, and also the nuclear envelope that you see, it has also disintegrated. Okay, started disintegrating. Now we have metaphase. We have the next phase, that is metaphase. So in the beginning of metaphase, the nuclear envelope completely disintegrates. So there it started off, but over here at the start of this, the nuclear envelope completely disintegrated. Chromosomes are spread in the cytoplasm. They are like, now I have a nice time. Okay, now chromosome condensation is complete. They are clearly visible under a microscope. Children, where did chromosome condensation start? There in the prophase. I told you it is not going to end there. Okay, it's still happening. I told you there is no clear distinction that is happening. Still, also the nuclear envelope started disintegrating there, but over here it have finally completed. Here chromosome condensation also have completed. Now the chromosome structure at metaphase. What is happening? So each chromosome is equals to 2 sister chromatids. Okay, are together held by what? A centromere. Are together held by centromere. Kinetochores, that are small disc-shaped structure, are present on the centromeres. Oh my God, what all things are happening over here? So the cell which was there, my dear children, the cell which was there over here, I'm saying that they would be having chromosome which is attached, sister chromatids which are attached with the help of a centromere. This is what? Centromere. And also there will be disc-shaped thing over here which is known as what? Kinetochore. Which is known as kinetochore. You know why there is a need of kinetochore? Because the spindle fibers which are going to come, okay, from the opposite poles, my dear children, they say I will be only attaching to the chromosomes if there is kinetochore. Otherwise, I'm not going to attach. So many people think that it is attaching to centromere. No, it is attaching to something else that is known as kinetochore, which is a small disc-shaped structure that is there. Okay, so this is the structure of chromosome over here. Okay, spindle fiber attachment. Spindle fibers attached to the kinetochores. Imagine that the spindle fibers would be coming and attaching over here. Each chromatin connects to the spindle fibers from opposite poles. From the opposite poles. So from the opposite poles, both of them are going to get attached. Each of them. Now alignment of metaphasic plate. Okay, so that is done. Chromosomes are moved to the spindle equator. They align along the metaphasic plate, perfectly lined for separation. So there is a metaphasic plate over here, my dear children, and what is going to happen over here? Here the chromosomes are going to come and line up like assembly, no? Like that. They are gonna line up over here, and you know who made them line up? The spindle fibers made them line up. So each chromatid is attached with one spindle fiber. With what? One spindle fiber. And where is the point of attachment? What do you call this as? The point of attachment as? You call this as kinetochore. So if you are asked how many number of spindle fibers are there, what will you answer? How number of spindle fibers are there? What will you answer? So I am saying 46 number of chromosomes are like this. There are 46 number of chromosomes. So how many number of spindle fibers are there, my dear children? 46 means 2 sister chromatids. Each sister chromatids are attached with one spindle fibers from both direction, my dear children. So what we do? 46 into 2. What is the answer? 46 into 2. Please calculate, my dear children, all these mathematics geniuses, because you know. No, and I am not saying. So 46 into 2 is basically 92. Okay, so 92 spindle fibers will be there at this point of time, just for you to understand. So in this phase, what all things are happening? Basically, nuclear envelope, it completely got disintegrated. The chromosome is finally happy, it's there. Okay, it's completely visible also now. Chromosome condensation has finally taken place completely. Okay, and now the chromatids are clearly visible and everything. You have a kinetochore also, that is the small disc-shaped structure that you have. And then basically, what is happening? The spindle fibers which are there, they have been coming together and they are attaching to the opposite sister chromatids through the kinetochore that is there. Now each chromatid connects to spindle fibers from the opposite pole, that is done. After that, they are like, come here, stand in the line. What is this line? Metaphasic plate. It is known as metaphasic plate or the equator. Okay, they are perfectly lined for separation. This is what happens in metaphase. So as you can see over here, they're like la 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 la, but at the end, they thought they were finally free. But again, they are asked to stand over here in one particular plate, that is known as metaphasic plate in metaphase. Fine. Now, going ahead. So number of chromosomes, my dear children, still 46. Still what? 46 only, because they are attached to the centromere. The chromatids are attached to the centromere. Okay, now anaphase. So beginning of anaphase, chromosomes at the metaphasic plate split simultaneously. Arre papa, again twist is happening. Each sister chromatid is now an individual daughter chromosome. What is the twist that is happening? The twist that is happening is that basically it was like this. they had a centromere. Now it will be like this. So when you have two centromeres, if this is one chromosome, how many chromosomes are over here, my dear children? These are 2 chromosomes now. This will be what? 2 chromosomes. Now finally they were becoming friends. They are getting disturbed now. So each chromatid is now an individual daughter chromosome because they are getting separated. Okay. And that's not all. Movement is still happening. Daughter chromosomes migrate towards the opposite poles. This is really bad, no? Centromeres lead the way while chromosomes arm trail behind. What do you mean by that? So basically, if you see, the spindle fibers are over here. So spindle fibers are attached on the kinetochore. Where is the kinetochore? The kinetochore is over here. Okay, the kinetochore is over here, that is towards the centromere. Now, just imagine like this. So, centromere, and so these guys, my dear children, these guys are like, I don't want to go, I don't want to go. So, but they still, like, the spindle fibers are pulling them apart. So it's like DDLJ, Dilwale Dulhaniya Le Jayenge. So in that, what is happening? Basically they're like, come, and the train is still going. Similarly, just like that also, here the hero and heroine are getting separated, my dear children. So, but the centromere which is there, it is going to what? Initiate the things. Yeah, it will be on the front side, and the chromosomes which are there, it will be towards the back side, which is why it is written that centromeres lead the way while the chromosome arms trail behind. Okay, now the key characteristics that you see over here, centromeres split, separating the chromatids. Yeah, the centromeres are splitting, separating the chromatids. Chromatids move to the opposite poles, ensuring equal chromosomal distribution. Ensuring what? Equal chromosomal distribution. So at this particular state, if I am asking you how many number of chromosomes are there, what will be your answer? So if it was 2n number of chromosomes from the starting until now, my dear children, at anaphase, if you talk of a cell as a whole, I can say it has double the number of chromosomes, that is 4n. Why? Why do I say so? Because, my dear children, they are finally separated. They are finally separated. If it was this was 1, it became 2. So what will 2 become? 2 will become 4. Get it? There will be doubling. There will be doubling of number of chromosomes. 92 number of chromosomes will be there over here. But are they going to remain at this stage, my dear children? No. Usually no one talks about it. Just for your understanding, I just told you that this is happening over here. Okay, so centromeres split, separating the chromatids. Chromatids move to the opposite poles, ensuring what? Equal chromosomal distribution. Now going ahead, the next thing, telophase. That is the last phase of your karyokinesis. That is a nuclear division. So chromosomes reach the opposite pole finally. Yeah, they decondense and lose individuality. What is happening over here? They were condensed to form chromosomes. Now again they get decondensed. Okay, they are in the older state, how we started off in prophase, my dear children, remember? So whatever is there in prophase, ulta of that happens over here. The nuclear envelope went over there, but the nuclear envelope will be reforming. Okay, around each chromosome clusters, 2 daughter nuclei are established. So basically, the chromosomes which were there over here, my dear children, where is it? Come on this side. No, it won't come. Okay, so I will go. You are not coming, I'll go. So basically what happens, around this a nuclear envelope comes, around this also a nuclear envelope comes. So again, my dear children, the cell we started off had 46 number of chromosomes, Here this cell also will have 46, this cell also will have 46. At the end, aren't you getting 2 cells with 46 number of chromosomes? Yes. So only in anaphase we can say basically this clear distinction is visible where 92 number of chromosomes is there in total, my dear children. Okay. And yeah, so in telophase, the formation of nuclear mammalian happen. 2 daughter nuclei are established. You get 2 daughter nuclei completely, and the chromosome also got decondensed. Reappearance of the organelles, all the guys that went on, my dear, okay, they all came. Nucleolus, Golgi complex, ER, come, everyone has come back. So you also have to come. So everything resets. Chromosomes settle down, nuclei reappear, and the cell gets ready for the final split. Okay, it's the final split that is going to happen. So until now, you were in the 30 minutes of your start of the examination. Now we are going to talk about the next 30 minutes, which is going to be really rigorous. Okay, so my dear children, I hope you understood karyokinesis. We have 4 stages. One is prophase, then is metaphase, then is anaphase, and then there is telophase. Okay, now we are going to get ahead. The nucleus has divided. The cell has to divide now. And cell division, what do you call that? Cell division, you call that as cytokinesis. Cytokinesis. Okay, until now karyokinesis, now we are going to talk about cytokinesis. So mitosis isn't over yet. After nuclear division karyokinesis, the cell itself must split into two. This final step is known as cytokinesis. So cytokinesis is different in animal cell and in plant cell. In animal cell, my dear children, do we have a cell wall? No, we do not have a cell wall. So it's like basically taking off a pizza slice. How is the pizza slice taken off? It gets ripped like that. Similarly over here also, basically what happens, or let's not compare it with the pizza also. Let's just drop it. Listen to me carefully. So in an animal cell, pinching happens. Okay, pinching off happens, as in a furrow is there. Okay, suppose this is a cell that you have. Imagine, use your imagination, my dear children. Everyone imagine. There's a cell. From between, it is getting moved inwards. It is getting moved inwards. This is known as cell furrow formation. Why is it happening? There are certain microfilaments up there, okay, inside the cell, which are pulling it inside. Which are what? Pulling. Suppose I am inside the cell and I am pulling from both the ends, my dear children. What will happen? The middle portion is going to get split like this. Okay, a furrow appears in the plasma membrane. The furrow deepens and pinches in until the cell splits into two. Okay, that is how cell splitting happens in the case of animal cell. Always remember, they have what? A cell furrow formation being done. Cell furrow formation being done. Ma'am, why did you tell about cell wall, by the way? If cell wall was there, do you think the cell furrow formation can be done? No. No one can make a furrow over there. It's too thick, the cell wall that is there. So I cannot afford to make a cell furrow over there. In the case of plant cell, things are quite different. In the case of plant cell, things are quite different. So what happens is that within inside, my dear children, the Golgi complex and lot of other things, basically they are going to make up something over here. And this something, my dear children, this something which is known as cell plate, later on it is known as cell plate. Initially there is a different name, I am not telling that to confuse you, but there is a cell plate formation happening. And what happens in cell plate, my dear children, because of the secretions being done by the Golgi patterns and all the other things, basically the cell plate is going to enlarge from both the sides. It is going to move towards both the sides, and when it moves towards both the sides, finally when it reaches the cell wall, it divides completely. It will be dividing the cell completely. So no furrow because a rigid cell wall prevents it. Instead, a cell plate forms in the center. The plate grows outward. Okay, the plate is growing outward. Remember, in the case of animal cell, it is growing inward. Over here, the cell plate is growing outward to connect the existing cell wall. Yeah, there is a cell wall out there, so it is going to connect it over there. The cell plate becomes the middle lamella separating two daughter cells. So usually, you know what, between the two plant cells that you are having, okay, there is a thing that is known as middle lamella. There is a thing that is known as middle lamella which is going to be formed. So cell plate becomes the middle lamella separating the two daughter cells. Sometimes cytokinesis does not happen. This results in multinucleated condition. You only think, okay, wait, let me explain it properly. Okay, sorry. So this cell, okay, so in the plant cells you have middle lamella. So what is this middle lamella, my region? Actually the cell plate which made it divide itself into two. Okay, now talking about, this is how I zone out. Okay, this is the usual way I zone out. People do not even understand that I have zoned out, but I understand. Yeah. So sometimes cytokinesis does not happen. If cytokinesis is not happening, my dear, what will happen? The nucleus is there. Imagine a condition, okay, over here. There are multiple nucleus over here. Cytokinesis has not happened. You will be obviously having a multinucleated cell. Yeah, more than one nucleus. Multinucleated cell will be there. What do you call this as? You call this as syncytium. What do you call this as? Syncytium. Okay, sometimes cytokinesis does not happen. This results in multinucleated cells forming a syncytium. That is liquid endosperm in coconut. If you remember, my dear children, coconut. Okay, in 12th grade you studied. In coconut, the water that we drink is nothing but actually a tissue that is known as endosperm tissue. It's a part of that. The cells, where the cells basically, the nucleus is keeping on dividing. The nucleus keeps on dividing. It's a free nuclear condition that is happening over there. So that is syncytium. Okay. Summary: animal cells pinch. Animals, you know, animals, they are pinching. This is not pinching, but yeah, animal cells, they would be pinching, but the plant cells, they are going to build. So plant cell build up our ecosystem, my region, where animals like us, not the other animals, they pinch. But sometimes cells just keep their nuclei without splitting, and in that case it is going to form what? Syncytium, and it will result in what? This is what? A multinucleated condition that you have. Got it? And you see that in the case of liquid endosperm of the coconut. Now, significance of mitosis. We started about mitosis. You should know about its significance, right? So first thing is growth booster. we organisms, suppose if mitosis never happened, we would be this much only. So mitosis has happened, which is why the cells are increasing, which is why we are growing, my dear children. Multicellular cells grow because mitosis creates what? New cell. And apart from that, nucleocytoplasmic ratio fixer. Yes, so suppose nucleus are way too much, cytoplasm is less. So you have to fix that, right? The nucleus and the cytoplasmic ratio, you have to fix it. How do you do it? Basically, we say that mitosis happens where each cell get one nucleus. So when cell gets too big, mitosis helps restore the balance by dividing it. The nucleocytoplasmic ratio is fixed with the help of that. Repair master. For example, I got myself hurt. So my stomach is making some noises. Probably I'm hungry. Yes. So anyways, over here, where was I? Yes, repair. So suppose I got myself hurt over here. Don't you think that it will be recovered? How does it recover? The cells which are there beneath my dear children, they are gonna basically not fight. They are gonna again do mitosis. Okay, they are gonna do mitosis, and the new cells would be replacing the old worn out cells out there. Okay, so lost skin cells, damaged gut lining, worn out blood cells, no worries. Mitosis keeps replacing them. I know I'm good at advertising. Yes. Now, plant growth engineer. Okay, so these are different terms that I use. Generally, I hope you understand that mitosis is important for growth, for maintaining the nucleocytoplasmic ratio fixer, and for repair, and also for helping in plant growth and things like that. So mitosis in meristematic tissues, apical and lateral cambium, helps the plant keep growing. If this is just about the plant, okay, so it is going to help the plant growing. It is also going to help the animal grow as well. Some haploid insects like male honeybee can also divide by mitosis, though it's usually restricted to diploid cells. So as I told you, my dear children, haploid, usually mitosis is done by diploid cells. We studied it right earlier in the case of animal cells. But in the case of honeybee, okay, which already has 60 number of chromosomes, usually it should be 32, but it just has 60 number of chromosomes. This is haploid condition. This is 2n, this is n, that is half of it. That is known as haploid condition. At this condition also, they will be undergoing mitosis to actually form their gametes. Okay, so that is what is said over here. Some haploid insects like male honeybees can also divide by mitosis, though it is usually restricted to what? Diploid cells. So in short, mitosis is equals to growth plus repair plus balance plus endless plant growth. Not just plant growth, all the growth. I hope that's clear. With that, we complete mitosis. Children, my dear children, please do not be worried about the questions. I would be coming up with questions also of these things. Chapter by chapter, I am figuring it out. And please make sure you are prioritizing your PU2 and you have studied all the PU2 chapters really well, I'm telling you again. So those of you have completed at least 6 chapters of PU2, the major chapters, do comment down that I have completed 6 chapters, ma'am. I am in progress. Okay, I would be really happy to know that. So next is meiosis. I'll take a break. I'll come back with my notes and I'll start off with meiosis. You also please take a break. Go back to all the things that you have studied until now. Okay, do not listen to me again. You will get bored. I know that. I'm not that interesting. But anyways, okay. So you have to use the slider and move through all the slides and read what is written over there. So you'll get an idea of what you have studied. Okay. And until then, I'll be just back. Please drink water and stay hydrated. So are we ready to go ahead with meiosis? I hope we are. I fully trust in you, my dear children. I hope you have done with the preparation for mitosis. You went back again, read through each and everything. Now you are not gonna get confused between meiosis and mitosis. Chalo, let's start meiosis. So here, general thing about meiosis, given that it happens in gametic cells, I told you already, right? Why in gametic cells meiosis happens? Because the gametes should be having half the number of chromosomes, and meiosis is the division that give you half the number of chromosomes at the end. So basically, for example, I start off with a cell that is having 2n number of chromosomes. Okay, when I end up meiosis, I will get cells with half the number of chromosomes, and this is very much important in the process of what? In the process of sexual reproduction, because n from mother, n from father comes together to give back what? 2n. So this is a general idea that we have, which is why we call meiosis as what? Reductional division. What do you call it as? Reductional division. In the case of mitosis, you used to call it as what? Equational division. Equatorial division. Equational division. Yes, because it is giving you equal number of chromosome that you started off with, or the number of DNA that you the amount of DNA that you started off with. Now, my dear children, over here there are certain things written. Meiosis involves 2 sequential cycles of nuclear and cell division called meiosis I and meiosis II. What do you mean by that? we studied in mitosis, there is only one thing, that is the cell is there, the cell divides into two. But here in the case of meiosis, there are two stages. In one stage the cell divides into two, just like this. But here halving of number of chromosome happens. Halving of number of chromosome happens. This is known as meiosis I. Then again the cell divides into two. This also divides into two. There is no halving of number of chromosomes, so hence it becomes n only. There is only increase in number of cells. So this is known as meiosis II. Yeah, and this is exactly like mitosis. Okay, in mitosis you know there is no reduction. Similarly here also there is no reduction. But anyways, when we say one meiosis has happened, that means two stages of meiosis have taken up there. Meiosis I and meiosis II. Now, meiosis I is initiated after the parental chromosomes have replicated to produce identical sister chromatids at S phase. So it is said that meiosis I, when does it happen? Basically after your chromosomes, my dear children, basically the DNA which is there, it has replicated. Now you have one more strand over here because there is a copy of DNA being made. This is known as what? Sister chromatid. All of it we studied already, right? So after this particular thing, after the S phase, there is G2 phase, and after that you have meiosis I being started off. Now meiosis involves pairing of homologous chromosomes and recombination between non-sister chromatids. There are so many things formed over here which might confuse you, which is why I will talk about this point later on. Now 4 haploid sets are formed at the end of meiosis. I told you one cell divides into two. This 2 cell again will divide into 2. So 2 and 2, you get what? 4 cells. 4 of them are going to be what? Haploid. They would be having half the number of chromosomes. So if we started off with a cell, if we started off with 4n, at the end we will be getting the cells with 2n, half. Okay. If we started off with 2n, we will be getting n. If we started off with 6n, we will be getting 3n. Anyway, it is going to get half at the end of meiosis. Okay. And usually we get 4 cells at the end of meiosis. Now meiosis I, that means that is prophase 1, metaphase 1, anaphase 1, telophase 1. Just how we had it there, right? Prophase, metaphase, anaphase, telophase. Just like that, we have 1, 1, 1, 1 over here and 2, 2, 2, 2, 2 over here. Now we are going to talk about prophase 1, and this is the most important part of meiosis. Okay. So prophase 1, my dear children, again is divided into certain stages. One is leptotene, then is zygotene, then is phycatine, then is diplotene and dikinesis. Okay, what is it? Leptotene, zygotene, phycatine, diplotene and dikinesis, LZPDD. If you have a fun way of remembering it, do tell me in comment section. There should be some mnemonic around it. I'll also share my mnemonic probably. Do you want it now? If you want it now, just wait for a second because I forgot. Yes, I am back, and let me tell you, due credits goes to Google, not me. So let zebra play during day. That is the thing. Let zebra play during day. You can remember it like that. Okay. Now, my dear children, what happens in all the stages? So usually what happens in prophase, my dear children, you know the chromatid which is there. The chromatin fibers which are there, it get condensed to what? Chromosomes. That is what usually happens, and the nucleolus, everything disappears, and there are a lot of things happening. But here there is little spicy game. Okay, apart from all this happening, there is other things also happening in the background. So let's see what are those other things. First thing, in leptotene stage, it is known as chromosome spotlight. As in, the chromosome becomes visible under the microsome and the DNA compacts like a suitcase before a long journey. So over here, I can say that the chromatin condensation will be starting off, and the chromosomes would be starting to get what? Visible under the microscope, my dear children. Now comes, and the DNA, it becomes compact, because obviously when you say chromatin is getting condensed, that means obviously DNA is being compact, which is why you have the chromosomes being visible there. Now you have zygotene. Zygotene is a stage where everything gets partnered up. Okay, so homologous chromosomes start pairing up. What do you mean by that, ma'am? So basically, we say we have 46 number of chromosomes. I don't want to make this area bad. So we say, children, we have 46 number of chromosomes. Agreed? Agreed? Cool. Now 46 of them, out of that 23 you receive from your papa, 23 you receive from your mama. Okay. So 23 each. Now basically, one chromosome from each. For example, I received first chromosome from papa, another first chromosome from mama. This will be 1 and this will be 1A probably. Again, the same thing happens, 2 and 2A. You get it? You receive both of this from both of your parents, my dear children. Now, when these guys come together, because these guys would be having same bands, gene bands, same type of genes and lot of things, my dear children, which is why I'd be calling them what? The homologous chromosome. They would be having genes coding for the same character. As in for example, here I have a gene coding for height. Here also I would be having a gene coding for height. Now it can depend. It can be a recessive allele over here. It can be a dominant allele over here. Anyways, I would be definitely having a gene that will be coding for height there. Whatever it is, depends on the parents. Okay. So anyways, it will be there. Now what do you call these chromosomes as? You will call these chromosomes as homologous chromosome. What do you call this? Homologous chromosome. So over here, homologous chromosomes start pairing up. So I want you to remember that basically, if you remember, my dear children, it will be really good for you only. So we have chromosome like this. This is one chromosome. Now, because it has got duplicated now. Now we have another chromosome also coming up like this. This will be what? The homologous chromosome. This will be what? The homologous chromosome. Okay, that I have. They would be coming up. Okay, so homologous chromosome, my dear children, over here, they are going to start pairing up. So if you have 46 of them, okay, you have 46 of them, how many pairs will you find? How many pairs will you find? 23. 23 pairs, right? 23. 1 comes together, you form a pair. Now 23, but the only difference is that since it has undergone duplication, now it looks like this. So do not take it to these are 4 chromosomes. No, they are just 2 chromosomes, my dear children. So we have 23 pairs of them over here, 23 homologous pairs over here. Cool. Now they are pairing up, and this process is known as synapsis or synapsis. Now the thing is that they don't want to be paired. So which is why we need to put some glue in between. We need to put some glue in between, and this glue is known as synaptonemal complex. What do you call it? Synaptonemal complex, which is going to hold these guys together. Now the paired chromosomes would be now known as bivalents or tetrads. Okay, they would be known as bivalents or tetrads. My dear children, when the condition is like this, you call this as univalent. You call this as univalent. Okay, there is 2 sister chromatids attached to one centromere. You call this condition as univalent condition. Okay, and you also call this as dyad. You also call this as dyad because you have 2 of them coming together. Now, if similarly you have one more chromosome like this, my dear children, this and this coming together, you will now call this as bivalent. You will now call this as bivalent, and it will be what? 1, 2, 3, 4. How many chromatids are there? There are 4 chromatids, so it will be a tetrad. It will be what? Tetrad. So this is univalent, but now this has become what? Bivalent. And if you count the number of chromatids, it's a 4 number, so it becomes tetrad. So my dear children, at this particular stage, I can say that paired chromosomes are now called bivalents. These would be now known as bivalents, and they are also known as what? Tetrads. Cool. In leptoprotein, the chromosome becomes visible. Okay, the DNA compacts out, that is DNA condensation has started off over here, right? Or basically chromatin condensation has started off, which is why you get the DNA to be really compact. Now in zygote stage, the homologous pairs comes together. The homologous pairs come together, one from here, one from here. So how many pairs? 23 pairs will be there. All of them are going to come together, and this is known as synapsis. But for them to stick together to each other, you need something that is known as synaptonemal complex that is being formed over here. Now, since they are all together, my dear children, I will call this as bivalent or tetrad. This happens in zygote. This happens in what? Zygote. Now next thing, pi-chitin, the swap meet, okay, they are going to swap something up. So the 4 chromatids of each bivalent become distinct. It will be very much clear, it will be very distinct. Recombination nodules appear, that is sites for crossing over, my dear children. So what happens is that basically now since they are together, they are going to produce some recombination nodules with the help of which they are going to exchange their things. They are going to exchange their things between them. So what's crossing over? A genetic exchange between homologous chromosomes. What is this thing crossing over? Genetic exchange between homologous chromosomes, my dear children. But before getting into that, I want to tell you one more very important thing, and that is these two chromatids that you have over here, aren't they sister chromatids? They are sister chromatids. What about them? They also are sister chromatids. Now when you talk about both of them, what about both of them? They are non-sister chromatids. They are not sister, right? They are non-sister chromatids, but of what? Homologous chromosome. But of what? Homologous chromosomes. Got it? Please remember that term. So what is going to happen is that there is genetic exchange that is going to happen between the non-sister chromatids of homologous chromosome. Cool? So they are going to exchange something. So the yellow part, I'm going to make it over here. So it becomes a bit yellow over here and it becomes a bit white over here. So there's an exchange that has happened over here. Who's the boss here? An enzyme called recombinase. So enzyme which is known as recombinase will be helping in this genetic recombination, which is also known as what? Crossing over. So what is 5-chitin? 5-chitin, basically there is a swap happening over here between the chromatids which are there. So recombination nodules are being formed, and with the help of recombinase enzyme they are going to swap certain things, and this is known as crossing over. So 5-chitin for crossing over, zygotin for what? Zygotin for synapses. Now diplotin, what happens in diplotin? Breaking up is hard to do. So something like that is going to happen. Synaptonemal complex dissolves, my dear children. This complex which is there, it is now going to slowly dissolve over here, yeah, but chromosomes stay connected at crossover points. Now they are going to stay connected at what points? At the points that crossing over has happened. So crossing over happens, they have not separated because they are connected over there. You get it? They have crossed over, but they are kind of connected over there. And this forms what? Chiasmata, the X-shaped structure. So if you see, so when they exchange certain things, my dear children, there will be an X-shaped structure that is being formed over here. So synaptonemal complex, it dissociates, but what happens? Still, even at a breakup, no, sometimes it will be like, we'll patch up. At that point of time, still, the chiasmata is basically holding up together. Okay, the crossovers points at there, you form chiasmata which are X-shaped structure which is still there. In some species like human oocytes, this phase can last for years, my dear children. It can last for what? Years. Please remember, what is the example? Human oocyte, that is the human egg cell. Okay, here in this stage it can remain for years. Okay, now that is about diplotin. Diplotin, basically synaptonemal complex goes. Now you have what? Chiasmata, which is the X-shaped structure that is being formed. Now Dikinesis, what happens over here? So chiasmata move towards the chromosomal end, which is known as terminalization of chiasmata. So basically when they are trying to move, it's not like they will be sticking together, no, but they try to move. Okay, the breakup is still happening, my dear children. So what happens? The chiasmata is going to move towards the end region. So it is known as terminalization of chiasmata. Chiasmata move towards the chromosomal ends. Now chromosomes are fully condensed by now. They started off there, but now they are fully condensed, and the meiotic spindles are gonna form. The spindle fibers which are there, they are also gonna form. Nucleolus disappears, nuclear membrane breaks down, ready for metaphase 1. Ready for what? Metaphase 1. Finally, I'm just going to revise. Listen to me carefully. Okay, no drawing, just hearing me. Okay, so first thing is leptotin. The chromosome has started condensation, that is chromatin has started condensation. You get chromosomes. Chromosomes are kind of visible, okay, and it is undergoing condensation, okay, and the DNA also is becoming compact. Now comes zygotin. Let zebra play during day. So zygotin is where you get the first thing, that is synapses. That is synapses or synapses. Okay, so over here basically the homologous chromosomes are going to come together with the help of synaptonemal complex. They are going to stay together and form a structure which is known as synapses. This whole process is known as, not a structure, the whole process is known as what? Synapses. And now they are going to form a structure which is known as what? Bivalent. Okay, they will be known as bivalent, and they will be having 4 chromosomes, which is why they would be known as what? Tetrads. Okay, just give me a second. Okay. Okay, so yes, then we have pi-chitin, where what is happening, my dear children? The 4 chromatids which are there over here, they are going to start forming what? Recombination nodules, and they will be crossing over, they would be exchanging their material. And here the major enzyme that is involved is what? Recombinase. Okay, now the next thing is diplotin, where breaking up is happening, my dear children. The synaptonemal complex which is there, it dissolves, okay, and then but still they are kind of linked together with the help of what? Chiasmata, that is X-shaped structures that will be there. Now, in dikinesis, okay, it is, okay, you can remember like that. Dikinesis, they are about to die, okay. It's not possible. It's not possible for them to basically, should I be telling it? Should I be not? I should not. Okay, let's just drop that idea. Anyways, in dikinesis, chiasmata basically move towards the chromosomal ends, and this is also known as terminalization of chiasmata. And since the whole prophase is ending, my dear children, the chromosome becomes fully condensed, and the meiotic spindles, they appear, the nucleolus disappears, and the nuclear membrane which is there, that also breaks down. For the next thing, that is what? The metaphase 1, where basically the spindle fibers have to get inside and join to each of these guys that you see over here. Cool. So this thing happens. I hope this is extremely clear to you guys. Now going ahead with the next thing, that is metaphase 1, anaphase 1, telophase 1 and interkinesis. This is really easy. Listen to me carefully. So what happens in metaphase? So the bivalents which are there, so over here, these guys and the other guys, they are going to, so these are the homologous chromosomes that we have, right? These are the homologous chromosomes that we have. And please remember, my dear children, the link is still there. The link, they are still linked with because of the chiasmata being formed, okay? So they are going to arrange in the metaphasic plate like this. They are going to arrange themselves in the metaphasic plate like this. The spindle fibers are going to attach over here and over here. Similarly over here, over here. Okay, how many spindle fibers will be there? You don't need to calculate that, but you only think, we have 23 of them like this. We have 23 of them like this from both the sides you are getting. So 23 into 2 is basically what? 46 of them will be there. Okay, that is quite different from what you saw in mitosis. So bivalent chromosome, that is homologous pairs, neatly align on the equatorial plate like a marching parade, and then microtubules from the spindle fibers attached to the kinetochores of the homologous chromosomes. So it is going to attach on what? The kinetochores which are going to be present over here, there it attaches, just like how normal metaphase happens. But over here there's only one difference. There in the case of mitosis, there was only one chromosome which was there, but here you have 2 chromosomes which are aligned together. So there it was like 46 of them aligned like that, but here it is what? Just 23, because we have made the other 23 come upside. Okay, I hope that's clear. So microtubules from the spindle fibers attached to the kinetochores of the homologous chromosome. Now in anaphase 1, the big separation happens. The homologous chromosomes pull apart and move to the opposite poles. It's like best friends going to different colleges. My dear children, here I want your attention. So basically what is happening, these guys are gonna move this side, these guys are gonna move this side. So over here you would be having things like this. Agreed? Remember, they have exchanged, but I am not coloring it again, but they have exchanged certain things, okay? They definitely have exchanged certain things. So at this point of time, the homologous chromosomes have moved apart. If you are asked what is the number of chromosomes over here, what will be your answer? We started off with 46 number of chromosomes, right? We started off with 46 number of chromosomes. If I say 2n, what is the condition over here? Still it is 46 only. 23 move this side, 23 move this side. Still it is what? 2n only, my dear children. In this condition, it is still 2n. Okay. And what about the DNA? What about the DNA? DNA is also like that. It will be 4C. It will be 4C. Right? Because initially we had 2C. Suppose if we had 2C, it had duplicated and now it became 4C. So it's still like that in the whole cell. I'm talking about the whole cell. Now, my dear children, what will happen in the case of telophase? In the case of telophase, so anaphase, if you read, homologous chromosome pull apart and move to the opposite poles. But wait, sister chromatids stay together. Yes, the sister chromatids which are there, they are here together, not like that. Okay, there the sister chromatids were getting changed. They were getting separated, which is why the number of chromosome was increased. But over here, the number of chromosomes still remains intact. Why? Because the sister chromatids are still there. Okay. Now, in telophase, what happened? The nuclear membrane and nucleolus reappears. Cytokinesis follows, forming two haploid cells. My dear children, how have it became haploid? So, over here, if I consider this as a cell and this as a cell, you tell me, here you have 23 number of chromosomes, here you have 23 number of chromosomes. So, we started off with 46, but don't you think it has become N and N? So, we said 2N. We said 2N. 2N means basically two cells. When you divide, you get N and N. We started off at 2N and now we have two cells with what? N and N. I hope you understand the difference between mitosis and meiosis now. There when we were doing, what was happening is that 46 chromosomes were there. Okay, 46 chromosomes were there. In that 46 chromosomes, my dear children, one cystochromatid went over here, another cystochromatid came over here. So from 1 you got 2 already, you doubled it already, and then it underwent division. So basically it will be 46 here and 46 here also. So from 1 you divide into 2, but here it is not happening. The cystochromatids are still together. Which is why when you divide you get N and N. If you go back to that area, you will still understand it more clearly. You will see that difference more clearly. So basically in telophase, the nuclear membrane and everything reappears, my dear children, and then you have cytokinesis, the cell division, where at the end you will get two haploid cells, and you can also call it as dyad of cells. Dyad of cells as in two cells you are getting with haploid nature. So chromosomes slightly relax but don't return to fully stretched interphase state. So it is basically, its chromosome have slightly relaxed, but it has not been to the older way it was. Okay. So here ends what? Your meiosis I, and at the end of meiosis you get two cells with haploid number of chromosome. Now in between meiosis I and meiosis II you have something like this, which is known as interkinesis, the short breather. So a brief pause before meiosis II starts. No DNA replication happens because it already doubled before meiosis II. Okay, there is no extra DNA replication that would be happening. Please remember, it's a very important point. DNA replication happens only in the S phase. Once it enters the meiosis, it is not going to undergo replication again. Okay, cool. Now, we are going to deal with those cells which would be having what? Haploid number of chromosome. How many? 23. So, 23 will be looking like this. All 23 would be looking like this, my dear. There will be 23 chromosomes. And now, they will be undergoing what? Meiosis II. Now, meiosis II, what will be happening? You have prophase II, metaphase II, anaphase II, telophase II, just like your mitosis. There we started off with 46 number of chromosomes, but over here we are starting off with a cell which has what? 23 number of chromosomes. That is N number of chromosomes, my dear children. So in this case, what will happen? Again, in prophase, the same thing happens. Okay, the same thing happens. The nuclear membrane disappears and things like that. So happens immediately after cytokinesis of meiosis I, chromosomes stay compact and do not fully elongate. The chromosomes basically, they remain compact, okay, they do not fully elongate. Nuclear membrane disappears by the end of this phase. There will be again the nuclear membrane, because in the telophase you got back the nuclear membrane, right? But here it again goes away. Now in the metaphase, all these 23 chromosomes with the cystochromatids are going to line up over here in the metaphasic plate, right? Spindle fibers are going to form. Right, spindle fibers are going to form. In anaphase, what will happen? Centromere finally split. The centromeres will be splitting. So in this condition, my dear children, you get one chromatid over here, another chromosome over here, this chromosome over here. The chromatids have split. So if it was N over here, okay, if it was N over here, what is the number of chromosomes over here? If it was N over here, what is the number of chromosomes? Baba, they have divided. The centromeres itself have divided. So it becomes what? 2N. It becomes what? 2N if you talk about the whole cell. If you talk about the whole cell. But not to confuse you, my dear children, just to make you understand, it is 2N now. In telophase, what will happen? In telophase, the cell will divide. The nucleus, not the cell, the nucleus will be dividing. When the nucleus divides, it will be N and N. So you started off with N and again you are getting N. Just like how you had in mitosis. In mitosis, there was 46 number of chromosome, right? There was 46 number of chromosome. And then, so 46, in that case, I called it 2N condition. Then over here, when this happened, all the 46, chromosomes, they split. It became 92 now. It became what? 92 of them. They doubled. So it was basically 4N over there. at that particular point of time. And then later when telophase happened, my dear children, when the nucleus finally divided, you get 2n, 2n at the end. Similarly over here, you started off with what? N. You started off with n, here you got 2n, and again when telophase happened, you got n, n, n. Same thing happens in mitosis, okay. So chromosomes are re-enclosed in nuclear membrane, cytokinesis follows, resulting in 4 haploid daughter cells. This is known as tetrad of cells. At the end you get 4 cells, right, which is why you call this tetrad of cells. In the end of meiosis I, you say dyad of cells because you get 2 cells. I hope this is completely clear to you guys. So please remember these things, that is, this is the most important part of meiosis. And then comes this thing, meiosis II is just like mitosis, and this will be giving you tetrad of cells. Please remember that this gives you dyad of cells. Anything else that we have? Nothing else we have. Remember, in anaphase I, sister chromatids stay together. Only homologous chromosomes, they separate. Okay. Now we come to this line. Meiosis involves pairing of homologous chromosome and recombination between non-sister chromatids of homologous chromosome. Yeah, 2 homologous chromosomes, when the exchange is happening between these 2 chromatids, we will call this exchange between non-sister chromatids of homologous chromosome. Yes. Now it's totally clear. So meiosis involves 2 sequential cycles of nuclear and cell division called meiosis I and meiosis II, but only a single cycle of DNA replication that happens in the S phase, not after that. Okay. Now meiosis I is initiated after the parental chromosome have replicated to produce identical sister chromatids. At the S phase, after that only it will happen. Meiosis involves pairing of this. I just read it. 4 haploid cells are formed at the end of meiosis II. That's all. And now the significance of meiosis. What is the use of meiosis? It maintains the chromosome number across generation in sexually reproducing organisms, my dear children. If halving is not done, what will happen? The number of, for example, I get 46, I get 92 number of chromosomes in my cell. I do not need that. So I have to maintain this balance. The chromosome number should be there, maintained, and that is done by with the help of what? Meiosis. Now, it reduces chromosome number by half to ensure gametes have what? Haploid sets. It restores diploid number after fertilization. So this step only they have put in. Let's say if you combine this, you get this. So reduction happens and then restoration happens with the help of meiosis only. Because of meiosis only this thing can happen, right? Introduces genetic variability through recombination and crossing over. Yeah. So crossing over is happening, which is why we get new genetic varieties. That is why we are not exact replica of our parents. Otherwise, basically 2 of them come together, then we should have been looking like both of them. But why is there some changes in us? First of all, because we are getting 2 sets, and apart from that, there is also crossing over happening, which is why we are forming new combinations as well. Now, variation, can lead to what? Variation, and variation is what? It is the one that drives evolution, my dear children. Now, the better ones would be adapted. Obviously, if you have a bad variation, that could be dangerous for you, but if you have a good one, that would let you adapt and survive better. So, these are the significance of meiosis. Please understand that. Okay, that is the only thing that I want to say, and yeah, that's all. That sums up. Children, I understand that you might have doubt at this particular area, but if you feel that I explained you in a better manner. Oh, sorry. Oh my God, my head is actually exploding inside. With all this information, but I hope I was able to explain you. If you feel you need more clarification, please feel really free to ask me in the comment section. I am here to help you guys. Okay, so only difference between the meiosis and the mitosis thing comes over here, where the homologous chromosomes are getting separated in meiosis, whereas there it was just the sister chromatids that were getting separated in mitosis. So in meiosis also sister chromatids get separated, but in meiosis 2, not in 1. And that creates the whole difference, my dear children. So if you found this particular video to be useful, definitely comment it down and do share it with your friends. Definitely do not forget to like the video as well. Wishing you all the best for your KCET examination. Bye-bye. Take care. Lots of love.